Hi, I'm Zoe Nielsen, the founder and president of Kennedy Capital. We are a small to mid-cap growth accelerator based here in San Francisco. And today we have two guests, Trent Hayes, who is the president of Kennedy Bio, which is one of our teams, and Eric Penney, who is a consultant and our FDA expert, one of our FDA experts, but I'll let Trent maybe give just a 2-second bio, and then Eric will hear from you.
Sure. Yes, hi, I'm Trent Hayes. I have a journeyman in the lab industry. I've owned several laboratories. I've done mergers and acquisitions with several of them. And I also helped build laboratories from the ground up to nice exit strategies. Along the way, I've met a lot of great people. One of them is Eric Penney. So we welcome him to our talk, and he's definitely a COVID expert in today's realm regarding laboratory testing, biobank collections, pretty much the full gamut. So without further ado, we'll welcome Eric Penney.
Well, hello, and thank you for having me, Trent. I've worked in the laboratory industry for well over 20 years, and I typically help to scale laboratory operations so that they're a lot more profitable. We have dealt with Six Sigma consulting and other types of more technical consulting throughout the years. And when COVID hit, we decided that we were going to go in that direction so that we could supply the industry with the needed answers that they were seeking. So that's where we are today.
Thank you both. So today we're going to chat about maybe just do some market commentary on Elon Musk and his COVID test. I think it's shining light on, the quality, the availability of the testing. As you may or may not know, he received 4 tests, same test manufacturer, same nurse, and received 2 positive, 2 negative. And I think you two definitely have insight about why that is. So we'll just walk through a couple of questions and maybe give some insight into what's happening in the medical diagnostic space with regard to COVID. So first of all, I'd love to hear, I don't know who would like to field this, but Trent, when I was talking to you and I said, oh, did you hear about Elon? He received positive and negative test results. You laughed. So maybe you can just give a quick synopsis of why that, not laugh, but you said, oh, it's because this isn't this, right? So maybe you can just give a little bit of insight about the type of testing, the antigen versus the PCR, and why the kind of pro cons there, and why what we're seeing as far as, Eric, maybe you can address what we're seeing as type of all the testing that's going out there because you're working directly with the Warp Speed project. So Trent, I'll hand it to you to just kind of give some insight on Elon's experience with his testing.
Yeah, so in the world of testing right now, there's obviously multiple different platforms. There's PCR testing, antigen testing, antibody testing, and it's very confusing for the consumer, which one to get. And they all work in different ways. They're all affected by different factors. Most laymen don't read the package inserts that come with these laboratory reagents, but if you do, you'll find out a lot of very interesting stuff, such as studies of being done with as little as 25 people or 100 people, and then studies being done with 2,000, 3,000. But as we know, the PCR testing is the best in the market, but the time frame it takes is the issue. So the rapid antigen test at 15 minutes, certainly help, but in cases like Elon, he's going to have different results, even though it's the same specimen, same nurse, same everything. It's the sensitivity and accuracy, all these factors come into play. But, like I've always said, I'm not really the expert. So I would definitely defer to Eric Penney, who is definitely one of the country's leading guys on this subject, because he's working with, the FDA and a lot of other companies daily doing this. So Eric, please take it away.
Well, the problem with the antigen test is across the board. It's not just with COVID, right? So think of a pregnancy test. It uses the same type of lateral flow assay to get the result. And not all home pregnancy tests are equal, correct? And if you think about that, the COVID test is the same. It's not going to be equal. So each of the ones that we have available are fairly accurate for an antigen test. However, if you test within the first 5 days of either getting symptoms or being exposed, your chances of your chances of actually having a accurate result are very, very high. When you get past 5 days, the virus sort of quits shedding quite as much, so it's not as detectable. So either Elon had a very, very small viral load, or he had the virus for many days before he got that antigen test. Across the board with all of the antigen tests, the false negative rate is 48%. So it's a little bit better than flipping a coin, but it's not quite as good as molecular-based testing or the PCR-based type test. The PCR test varies. There's almost 300 that they've authorized so far. The best has a lower limit of detection of around 180, whereas the opposite end of that spectrum has a lower limit of detection of around 600,000. So just that gamut of numbers there can tell us sort of how each test would perform and how we would expect those false negative or false positive results to appear.
Okay, so question, thank you. So there's 300 that have their EUA so far, which I'll have you explain a little bit about what that means in a second and kind of what's gone on in that throughout COVID. I think it's pretty interesting. How does, let's use me as an example. I'm feeling like I have symptoms. You have to get a doctor's order to get a test, right? Correct. So me as a consumer, there's 300 available. How does the trickle down work of the best tests getting to me as a consumer?
So you don't have a lot of choice in that unless you go out and seek a specific brand of test out. So typically you would go to a collection site that either the government or your local community has set up. When you go there, you're sort of at their mercy for whoever they're sending testing to. Most likely, if it is a government organized testing location, those tests will be sent to the state laboratory. The state laboratory has a very small capacity to run those tests. So they partner with larger labs that can handle that overflow testing. Nobody really knows what method they're going to use. They all know that it's going to be a molecular-based method, but they don't know which platform or which manufacturer is going to actually run that test.
Interesting.
So, yeah, it's like a flip of a coin, unless you specifically know that a lab is using X technology and you go to that lab specifically, you don't know.
Interesting. So it's really a two-step process, the collection and then wherever that collection goes.
That's correct. But, even the collection process can alter the results and create a false negative result all on its own. There's a lot of factors there. What we worry about is RNA degradation. So after you collect the sample, it's placed into a transport media, usually VTM or UTM. Neither one of those medias contain an RNA stabilizer. So the RNA starts to degrade as soon as that specimen is collected. The only way to stop the degradation is to add an RNA stabilizer to the mix, which no one has done yet, or you could freeze it at -70 plus, and that would stop the degradation of the RNA, but that's not a feasible method of transportation.
Correct. Interesting. Okay, so let's go back to the EUA. And I found it interesting just watching the EUA and not only the CLIA overlay. And I guess I've been a little upset about the various tests coming to market as far as, widespread diagnostics, if with the antibody test or the antigen. So maybe you, with your experience with the FDA and your background in clinical trials and whatnot, maybe you could just weigh in a little bit on kind of what's been going on and how you see maybe what happens after all these EUAs, like they have to go back to the process. So would love to just hear your thoughts.
Yeah, so, the EUA process has been around for a very long time. The FDA started that, I don't even know when, but it's probably before my lifetime, to be honest with you. They have that just for emergencies. So if you remember back to Zika, when Zika came out, nobody could run a test on Zika. So what did they do? They sent out an EUA requesting companies to create a solution for this. So with Zika, it was a lot more organized because there was only 2 or 3 companies that decided they wanted to take up the challenge. With COVID, it seems like everybody wanted to get in on the game. So that's what you have. You've got thousands of submissions requesting review, but at the FDA level, they only have about 100 reviewers. So it's a slow process, and they want everything to be as good as it can be for what we're working with. So they take their time and try to understand why we have false negatives and why we have false positives and all of the problems, right? So one of the issues right now that is sort of hindering the accuracy of testing is there's no way to correlate a viral load with an actual lab value. So typically with a PCR test, the values are reported in something called CT or CQs. Those do not correlate with symptoms or a viral load. So they have no way to set a standard for testing. Once they set a standard for testing and they better understand symptoms as they correlate to a viral load, we will see a lot more accurate tests coming out. As far as after the EUA is over, currently I anticipate the EUA to last 4 to 5 years for many reasons that I won't get into today. However, when that process is over, they will start pulling the EUAs from the market. They will give each company an opportunity to bridge over to a 510K program. The FDA currently is considering the de novo pathway or the Q submission pathway, which are 2 very different ways to get to the 510K. I believe that they will probably end up using the de novo pathway ultimately, which is a little bit more expensive and a longer process for everyone involved. However, it does look at the platforms more in-depthly, and it requires a little bit more study on the manufacturer side to pass the test.
Interesting. Okay, I just have 2 more questions. One, obviously, it sounds a little doomsday, right? As far as getting a handle on the testing, the healthcare workers. I mean, it's just a lot to handle. And I've also heard rumors that the virus mutates. Do you have any thoughts on that?
So it's mutated several times. The last wave that America's seen was a clade called D614G. And when it mutated into that specific formation, it took the spike protein of the virus and it made it, instead of a shape that it can't change out of, it's now able to morph that spike protein into whatever shape it needs to unlock your cell. So in the beginning, children couldn't be infected quite as easily. There's an ACE2 receptor gene that we all have. Children have less of those than adults. So originally the thought was there's less genes for it to unlock, so that's why children are safer. But when the mutation D614G came out, it was quickly noted that now those ACE2 receptors in children are susceptible. So it made the virus more virulent. It didn't make it any more symptomatic. So the death rates and the amount of symptoms are the same, but it's able to infect more efficiently and more people, right? So the more easily it's able to get around, the more it's going to mutate, because the virus just wants the same thing that we want. It just wants to live its life. And it can only live its life if it's constantly changing so that our immune systems are caught off guard. Mm. And so there's 2 other mutations that we're studying now. The latest one's out of Europe, and then there's one out of Denmark. And both of those are very interesting because it also affected the spike protein. They're so new, though, we don't have a lot of data to really look at and fully understand what the consequences of those 2 mutations might be.
Mm-hmm. Bit of a moving target.
It is.
Keeping the testing going, if it's changing. Let's talk about a couple things that are positive. So tell me about warp speed. I'd like the world to know that exists.
Sure. So Project Warp Speed was invented by the CARES Act. It is a part of the United States government Rapid Accelerated Diagnostics Program, or RADx. The Warp Speed program originally had, I believe, 24 contestants that were chosen that had new and unique technologies that were related to COVID or combating COVID. You know, some of the companies you've heard of, like Pfizer's on there, some you've never heard of, like PathogenDx, but all of those companies do have brand new technology that we've not seen yet, either on the vaccine side, the testing side, or the treatment side. And out of those companies, you know, we hope to find both a cure and a better way to test the masses. One of the problems with testing everyone today is supplies. We just simply don't have enough of those. So there are some companies on that Warp Speed program that have some very novel solutions to those supply issues, as well as the cost of testing. So I think in the next few months, you'll see the cost of the test go down tremendously, along with more availability. I think that the turnaround times will continue to be an issue because laboratory staff members and nurses and really the whole healthcare field has been getting the virus. So there's less of those people available to perform those duties now. And I anticipate through winter with dual infections of influenza and COVID happening, that it will somewhat overwhelm the system a little bit. But you know, we're a resilient nation, so I think that we can come together and really squash this out.
So tell me about the vaccine. What do you know? Obviously, it's very hush-hush because it's not finalized. And
correct.
And then there's, you know, 8 billion people to give it to. So tell us.
Well, the good news is there's 8 billion worldwide, right? But each country is pretty much doing their own vaccine program, and then the WHO is running a very large program as well. So there's each one of those countries has wonderful candidates that have the potential to create a solid vaccine, right? In America, Pfizer and Moderna are probably the most well-known. Pfizer will probably be able to produce a vaccine that's FDA approved or authorized by the end of the year. That doesn't mean it's going to be distributed by the end of the year. It just means that it'll be available for distribution. Probably right after that, Moderna's will come out, maybe March or April of next year. Each of those vaccines will be a 2-dose vaccine, not a 1-dose. But you know, the trial data looks really, really good. If you want to go around the world a little bit, several months ago, we heard about a Russian vaccine that sounded almost too good to be true because it just sort of came out overnight, and all of a sudden they're already administering it to healthcare professionals and the president's family and different things. That particular vaccine, I believe, is around 92% effective also. And it's also at the end of its phase 3 trials, meaning that it will be ready for distribution very soon also. So between those 3 companies alone, we've got the ability to basically do maybe a third or a quarter of the world. But there's a ramp period. So the ramp period will be probably around 6 months to a year, but that doesn't mean that they won't be vaccinating during that time. So as they start making lots of these vaccines, they'll start pushing those out to pharmacies, health departments, doctors' offices, different places that we've already identified that would be a good place for people and convenient for people to go to receive the vaccine. You'll also see mobile units going out, vaccinating rural communities, and there may be some large vaccination events at you know, stadiums and theaters and things like that. So you know, they're not going to take very long to get it out. It will come quick.
That's good news. Well, thank you. Trent, do you have any questions? Eric kind of stole the microphone.
No, as he should. You know, like I said, he's definitely one of the experts. But no, I mean, he pretty much covered all the topics that I think a lot of people would want to know about from a laboratorian side of view, and you know, basically in the front lines like Eric is.
Yeah. Well, Eric, thank you so much. I've learned so much today, and appreciate you hopping on to chat with us. And Trent, as always, thank you for your time as well. So have a good day, gentlemen, and we'll chat soon.
Thank you.
Thanks, everyone.
Take care.